Hello, and welcome to today's webinar. Before we get started, I'd like to go over a few items so you know how to participate in today's event. All attendees are in listen-only mode and muted during the presentation. At the end of the presentation, we will take questions. To submit text questions to today's presenter, please type your questions into the question box labeled Q&A at the bottom of your screen. We will read them aloud during the Q&A session after the presentation. Neil's webinars are intended for general information purposes only. The information in the following slides has been made available without evaluation or endorsement by Neil's. By providing this information, Neil's is not recommending any treatment to individual patients. Thank you everyone for joining this Neil's Community Education webinar on promising alternative and off-label treatments for ALS. I would like to introduce today's presenter, Dr. Richard Bedlack.
Hello, everyone. Thanks so much for taking some time out of your day to join us. I'm going to be talking today about a topic that's near and dear to me, promising alternative and off-label treatments for ALS. And just one hint on this first slide, none of the things that I'm going to talk about today are on that shelf. So these are my disclosures. I'm not going to sit here and read all of these, but I do work with a lot of companies. I don't think any of these disclosures are relevant to today's talk, but in case you want to go over them with a fine-tooth comb, they'll be on the video. I also have some disclaimers. As you heard, I just want to make sure folks know the information in this talk is for educational purposes only. It shouldn't be interpreted as specific treatment advice. The viewpoints that I'm sharing today are my own. And also none of the products that I'm going to mention are FDA approved for slowing ALS progression. So here's the outline of today's talk. I'll start off by telling you how I selected this list. I'll then move into my current top 8 alternative and off-label treatments, and we'll go through those one by one, and I'll tell you why I'm excited about them. I'll give you one slide that I think is pretty interesting, and that is the fact that the mechanisms of these 8 products overlap so much. I think it tells us something. And then finally, I'll share a few slides on how I guide patients in my clinic who want to experiment with new products. So to get into how I selected these products, you have to understand the ALS Untangled program. So early in my career, I recognized that people living with ALS wanted to try experimental things, perfectly reasonable. And unfortunately, there weren't enough trials or expanded access programs for them. So many of them were just buying and trying things that they saw on the internet. And there didn't seem to be a standardized way to work with people to try to help them make more informed decisions. And so I got together with some colleagues in Neil's way back in 2009, and I put this program together. And we never really had another program, we still don't, in any other disease that's quite like this. So we've had to invent this. And we've improved and refined it over the years. Basically, it consists of 3 parts: where we get our ideas. So those can come in through emails to any one of the ALS Untangled investigators. They can come in through one of my colleagues who might be a clinician at another clinic and was asked about something from a person living with ALS. They sometimes come through social media. Once we get an idea, we post it on the future reviews page of our website, www.alsuntangled.org. Now, there's so many ideas that we keep getting that we're probably never going to get to all of them. There's over 500 ideas on that future reviews page. And so we've had to come up with a way to prioritize them. And the main way we prioritize them is by votes. So if there's something on there that you really want to hear about, get on that future reviews page and click the vote button next to that topic. And then we try to prioritize our reviews based on the ones that have the most votes. So once a topic gets assigned to one of the ALS Untangled team members, they write a review, they crowdsource it across the whole rest of the team, which is now 130 clinicians and scientists from across 11 different countries. And then once all those comments are incorporated, it gets sent off to a journal for an independent peer review. And once we respond to that, it gets published in the journal. And all of our reviews are published through something called free open access, so no one ever has to pay to read what we write about. And you can find those reviews on the completed reviews section of our website. If English is not your primary language, we now have many parts of the website translated into both Spanish and Italian as well. So one of the sort of keys to ALS Untangled was coming up with an objective way to look at each product. Like, we don't want this to just be one person's opinion about it. We want it to be as scientific and objective as possible. And so we created something which is called the table of evidence. And it's really important for today's talk because the products that I'm going to talk about are based upon grades in 3 categories on this table of evidence. So for each product, we tried to learn about 5 things: the mechanistic plausibility. What is this product supposed to do? And does that make any sense in terms of what we know about biology, and in particular, the biology of ALS? Preclinical models. Was this product ever given to any cell or animal model that's relevant to ALS? Case reports. What are people living with ALS who tried this saying about it, good or bad? And can we find those people? Can we prove that they're real, that what they're saying is supported by objective evidence? Trials. Was this thing ever put into a clinical trial? And what did that trial look like? And finally, risks. What bad stuff could happen to you if you tried this particular product? And within each of these 5 categories, we assign a letter grade based on the specific information that we find. And just like back when we were in grade school, you want to get A's and B's, and you want to try to avoid D's and F's. But the products that I'm going to talk about today, we're looking at 3 categories: mechanistic plausibility. And so let's take a look at the grades there. So we're only going to consider products that have a plausibility grade of D or higher. So there's not going to be any products I talk about that have a mechanistic grade of F, because that means they're supposed to work in a way that would violate the known principles of physics or biology. And someday those might change and we could reconsider those products. But for now, your product has to at least act on a biological mechanism. If it's not clear that it's relevant in ALS, you get a D. If it is relevant in ALS, you get a C. If you've actually shown that your product hits that mechanism in a preclinical model that's relevant to ALS, you get a B. And if you've shown that it hits that mechanism in people living with ALS, you get an A. It's pretty rare to get a mechanisms grade of A. And then we're going to be looking at the trials category. And again, all these products have to have gotten a D or better. So we're not going to talk about products where the only trials that were done show no benefit. And so you'd get a D for a trials grade if you had your product in a trial and it showed a benefit, but the trial was significantly flawed. Like, for example, it wasn't randomized or blinded or placebo controlled. You get a C if there's one published trial that's pretty well designed, but it's small, like a phase 1 or phase 2. You get a B if there's one phase 3 trial, and you get an A if there's 2 or more published phase 3 trials. And then finally, we're going to be talking about the risks category. And again, you won't hear about any products that have a risks grade of F. That means at least 5% of people who got this died or were hospitalized. That's too high of a risk, in my opinion. And so you're going to have a risks grade of D or better. So D means 0 to 5% of people on this product experience hospitalizations or deaths. C means there were no hospitalizations or deaths, but at least 10% of people had some kind of side effects. B means no hospitalizations or deaths, and the percentage of people with side effects is 0 to 10%. And then A, which is quite rare, means that nobody ever had any side effects from this product. So to go into the full set of selection criteria that I use to pick today's products, first of all, these products have to be available. So if they're owned by a company and that company's not making these products available, they're not going to be on this list. They can't currently be labeled to show ALS, to slow ALS progression. I mean, if they're labeled to slow progression, they're not alternative or off-label products. All the products today have to have been reviewed or be actively under review, in other words, in draft status by ALS Untangled. So you're not going to see any products that are on the list, but we haven't gotten to yet, or any products that are not on the list of future reviews. All these products in their best available trial had to show a clinically meaningful benefit. That means, to me, it seems like something that would matter to a person with ALS. Now, sometimes these benefits reach statistical significance and sometimes they didn't. And the reason why that's not an absolute criteria is that a lot of the things I'm going to talk about, the trials are pretty small, probably underpowered to show a statistically significant benefit. So you got to have a trials grade of at least D, and the benefits have to be deemed to be meaningful. Next, you have to have a plausible mechanism of action, a table of evidence mechanisms grade of at least D, and then finally, your product has to be reasonably safe, a table of evidence risk grade of at least D. So that's the selection criteria. So here's my top 8 products as of today, and these are in alphabetical order. We'll talk about each one of these in a little bit more detail. Acetyl L-carnitine, antiretrovirals, clenbuterol, dextromethorphan quinidine, L-serine, methylcobalamin, nicotinamide riboside with pterostilbene, and tamoxifen. So let's talk about each one of these in a little bit more detail. So acetyl L-carnitine. Well, let's start with carnitine. Carnitine is an amino acid. It comes in a lot of different forms. You can get it as a supplement, and it seems to play a key role in cellular energy production by transporting fatty acids into mitochondria, where you can generate more ATP. This is one of the few products that actually has a mechanisms grade of A in the history of ALS Untangled, because it does increase mitochondrial ATP synthesis in humans. It also reduces oxidative stress in humans. And finally, there's some recent evidence that it might alter the gut microbiome. What about trials? Well, there is one trial that gets a grade of D. It's 82 patients. It's 48 weeks long. It was randomized, double-blind, and placebo-controlled. It used acetyl L-carnitine at a dose of 1,000 milligrams 3 times a day. And the main thing that it showed is that it kept people in what's called a period of self-sufficiency. So what that meant is they didn't need any help with feeding, dressing, riding, or walking. So those measures on the ALS functional rating scale were either a 3 or a 4, meaning you might be a little slower, but you don't need any help. And I think that most people with ALS would say that is important to stay self-sufficient. In addition, it slowed ALS FRSR progression by 35%. The reason it got such a low grade is because there's a lot of methodological problems with this trial. So 21 of the 82 patients had protocol deviations, including there were some patients enrolled who didn't even meet the trial's inclusion criteria. In fact, there were 2 that were later found to not even have ALS. So that's a problem. The good news is this is a pretty safe product. It got a risk grade of B. You can have rare, non-serious GI side effects, which is true of almost every supplement. And there are rare reports of a fishy body odor in less than 5% of patients. The other nice thing about acetyl L-carnitine is it's cheap. It's probably one of the cheapest products we'll talk about today. At the dose used in the trial, it's only about $15 per month. So product number 2, antiretrovirals. So this is a group of medications. They're all prescription medications, and they're used to treat retroviruses. So these products get a mechanisms grade of A. They do inhibit the life cycle of 2 retroviruses, namely HIV and HERV-K, that can cause upper and lower motor neuron deterioration. It's not widely accepted that either one of these is involved in the pathophysiology of ALS for most people. They also alter the gut microbiome. So here again, we've got a trial that gets a grade of D. Small trial, 40 people with ALS on a very specific antiretroviral, which is called Triumec. It contains 3 ingredients: abacavir, lamivudine, and dolutegravir. This is given once daily for 24 weeks. What the trial overall showed was about a 22% reduction in the rate of decline on the ALS functional rating scale. Also, there were some biomarkers that improved. You would expect that the level of this endogenous retrovirus, HERV-K, would go down, and it did. The other nice thing that happened is there's a biomarker of neuroinflammation called urinary P75 ECD, which improved. There was a follow-up publication reanalyzing some of the results of this trial, and it looked like 18 of the 40 people in this trial were defined as responders. Those were people who actually had a significant decrease in HERV-K levels, and their clinical responses were even better than people who didn't seemed to have a decrease in their HERV-K expression. So there's a lot of criticisms of this trial. It's very small. It's not randomized. It's not blinded or placebo controlled. But the good news is there's going to be a follow-up trial. In fact, I think it's enrolling. It's going to be a much larger trial, 390 patients, and it will be randomized, double-blind, and placebo-controlled. So soon we should have an answer as to whether this product is useful. In the meantime, Triumec does seem to be reasonably safe. Other antivirals have not been well studied in people with ALS. So really the risk grade is just based on Triumec. So in the trial, there were 7 adverse events, none of them serious, that seemed to be related to Triumec, 6 cases of nausea and 1 rash. Now, very, very important, and that's why I put this in red, everybody who was in this trial had to be screened with a blood test called HLA-B5701. So if there's anyone out there who's thinking of taking Triumec, the first thing is to get screened with this blood test. Because if you have this HLA-B type, that predicts an allergic response to one of the ingredients in Triumec. And so it would not be a good idea to take it. The cost is substantially higher than some of the other things on this list. Triumec at once a day is about $3,000 a month. Next, we have a product called clenbuterol. So this is a drug that's in a class of beta agonists. They're used to treat asthma in many countries. Clenbuterol is not used in the United States for any labeled indication. It's not FDA approved, but if you're a sports fan, you might have heard of it because it is used off-label by athletes to enhance muscle mass and athletic performance. And if you test positive for what they call clen, typically you get suspended or banned from your sport. It's considered cheating. Mechanisms grade is C. It actually has quite a few mechanisms by which it could plausibly slow ALS progression. And there's a nice review article that I've referenced there, but it can improve muscle protein synthesis, which is how muscles get bigger on it. It can stimulate neurotrophin release, improved mitochondrial energy production, and reduced neuroinflammation. So the best available trial for clenbuterol is the one that I did, and it gets a grade of D. Yep, I'm grading myself all the way down to a D, which is painful. But this was a small trial, 25 people with ALS, and we treated them with doses ranging from 40 micrograms a day, pushing it all the way up to the goal, which was 80 micrograms twice a day. And we had them on treatment for 6 months. Each person was their own control. So we compared different measures of disease progression for the period before treatment versus the period on treatment. And we did have a pretty significant dropout rate in this trial, 56%, which has probably partly to do with the dose that we chose, but also partly to do with the fact that we opened this trial the very month that the world shut down from the COVID-19 pandemic. And we had a lot of people that didn't want to come to clinic, understandably. And so there's different ways to analyze the results of a study when this happens. The most rigorous way is something called intention to treat. So everyone that started the study gets analyzed. And so if we use that rigorous way of analyzing the data, the main thing that we see is a really significant decrease in the rate of decline on the forced vital capacity, a measure of breathing. So we saw a 90% slowing in the rate of decline on clenbuterol. We also can look at the ALS functional rating scale. And if you look at the 20 patients in the trial who had at least one ALS FRSR measurement, 15 out of 20 had slower progression on this measure during treatment compared to before. We also look at hand dynamometry, grip strength, and also manual muscle testing. And we saw some patients in this trial get stronger over the course of treatment with clenbuterol, which in my experience is quite rare. So again, there's lots of reasons to criticize this. It's small, it's not randomized, it's not blinded, it's not placebo controlled. It's got a huge dropout rate. It needs to be replicated. So we gave clenbuterol a risk grade of C. And part of that is because even though we didn't see any serious adverse events related to it, we did have most people in this trial experiencing side effects from it. These included jitters, tremors, cramps, spasticity, and insomnia. Now, the frequency of side effects may have had something to do with the dose we chose. We were aware of an earlier case series from Italy where a neurologist just put 16 of his patients on clenbuterol at a much lower dose, 20 micrograms 3 times a day. And it seemed to be quite well tolerated, and there seemed to be benefits. We thought that if we pushed the dose even higher, it might be even more obvious that it worked. So if I were to be in charge of a replication study of clenbuterol, I would use 20 micrograms 3 times a day. And for folks who are trying to price that out, at 20 micrograms 3 times a day, the cost is about $270 a month. Now, again, you can't get a prescription for this and just go to an American pharmacy. It's not FDA approved here, but there is a way to get it. So there's a group out there called everyone.org, and their business model is they actually help people with rare diseases to import medications from other countries where they are approved into their country where they're not approved. And there's a whole process that has to be gone through, including getting a prescription from a doctor here in the United States, as well as a letter of support that has to be submitted. The next product on the list is dextromethorphan quinidine, which some of you may know as Nuedexta. So this is FDA approved for treating a symptom that we see in ALS, which is called pseudobulbar affect. This is where people with the disease lose control of their ability to regulate their laughing and crying, but it's not FDA approved to slow the disease. Mechanisms for slowing the disease, we give it a grade of B. It actually does several interesting things. It acts through a receptor called sigma-1R. And in that way, it actually is a glutamate antagonist. It modulates two neurotransmitters, norepinephrine and serotonin, and it blocks voltage-gated calcium channels, all of which may be involved in suppressing hyperexcitability in the brain. It also can reduce oxidative stress and neuroinflammation. So there is that second ingredient, quinidine, that I haven't mentioned. Quinidine doesn't seem to have any mechanism of action that treats ALS. So why is it in there? Well, if you just gave dextromethorphan, as it goes through the liver, it gets wiped out. It wouldn't be around long enough to get into the brain and do anything that might treat a person with a disease like ALS. So you've got to inhibit the liver's metabolism of it, and that's what the quinidine does. So there have been a couple of trials. The best one, in my opinion, gets a grade of C. This is something called a crossover trial. So here, 60 people living with ALS were randomized to receive either a placebo or Nuedexta. The Nuedexta dose was 1 capsule daily for 7 days, followed by 1 capsule every 12 hours. They were treated that way for approximately a month. This was followed by a washout of 10 to 15 days, and then they got the opposite of their initial treatment for another 30 days. So everybody in the trial got both a placebo and Nuedexta. They were randomized to see which one they got first. And so there was a pretty profound improvement in the Nuedexta treated group on a composite measure of bulbar function called the Neurologic Study Bulbar Function Scale, CNS BFS. So what are the parts of this? Well, it measures the clarity of speech, it measures swallowing, and it measures drooling. And not only was there a significant improvement on the measure as a whole, but each one of the individual domains of the measure also had a significant improvement. They did look at a more traditional measure, the ALS FRSR. There wasn't an overall improvement, but if they looked at the bulbar subscore, again, they saw a pretty profound improvement there. And importantly, these improvements occurred regardless of whether the person in the study had pseudobulbar affect. Now, again, this is not a perfect study. It's very small. It's very short. We don't know how long the effect might last. It probably needs to be repeated, with a much more traditional phase 3 design. This does seem to be a pretty safe drug. About 10% of people with ALS in Nuedexta trials have side effects, most commonly nausea, somnolence, dizziness, headache, fatigue, and diarrhea. You typically don't see serious adverse events with Nuedexta. But again, there's one thing to watch out for. Because of the quinidine, which is an antiarrhythmic, you do have to keep an eye on something called the QT interval. This is a part of the EKG tracing. If it gets too long, a person can actually have a fatal cardiac arrhythmia called torsades de pointes. So when you're thinking about taking or prescribing this medicine, the first question you have to ask yourself, is there any reason this person might have a prolonged QT interval? Like, for example, are they on another drug that prolongs the QT? Like an antidepressant. Many antidepressants prolong the QT. If so, what you need to do is a baseline EKG to make sure the QT is okay. You need to give a dose of Nuedexta in clinic, and then 3 hours later, repeat the EKG to make sure the QT hasn't prolonged. The cost of this is, again, fairly substantial. At a dose of 1 capsule every 12 hours, it's $1,500 per month. Insurance will most of the time cover it if it's for pseudobulbar affect, but not if you're saying in the notes that you're trying to improve speech, swallowing, or drooling. That is not a labeled indication of this medicine. Next on the list, we come to L-serine, another amino acid. It comes in two forms or enantiomers, L and D. These are widely available on the internet as nutritional supplements. You don't need a prescription for these. Mechanisms, it's got several, and it gets a grade of B for these. It blocks the incorporation of a neurotoxin called beta-methylamino L-alanine, BMAA, into proteins. This is a hypothesis about a cause of ALS. It reduces neuroinflammation by downregulating pro-inflammatory cytokines and microglial activation. It reduces markers of stress in a part of the cell called the endoplasmic reticulum, where proteins are processed. And here we go again, it changes the composition of the gut microbiome. There so far has only been one clinical trial of this in people with ALS, and it gets a grade of D. It's only 20 patients. And believe it or not, these were divided across 4 different groups. So you had 5 people get a very low dose, 0.5 grams of L-serine twice daily. Another 5 patients got 2.5 grams twice daily. Another 5 got 7.5 grams twice daily. And then finally, another 5 got 15 grams twice daily. And the ALS measurements that were made were compared to another study. They were compared to the placebo group of another study that had similar inclusion criteria. And what was seen was a trend towards slowing of ALS FRSR decline in people on L-serine. And it actually was quite impressive on the patients on the highest dose. Now, again, there were only 5 patients on that dose, but there was an 85% slowing. There was a replication trial underway in Dartmouth, but it appears that that trial has closed for administrative reasons. I don't have all the details on that. I was quite sad to see that, and I hope somebody will pick this up. Obviously, that one trial that's out there is very small, and it's not placebo-controlled. The risk rate is C, no serious side effects associated with this that I can find, but about 15% of patients have GI side effects. And when you get all the way up to 15 grams twice a day, that's 25% of people that can have GI side effects. I'd say this is modestly expensive at a dose of 15 grams twice a day. The dose or the cost can be $350 a month. Some of that depends on the supplier. Next on the list, we come to methylcobalamin. This is an active form of B12. It doesn't need to be metabolized in any way to do all the good things in the body that B12 does. This is the only product in the history of ALS Untangled that gets both a mechanisms grade of A and a trials grade of A. So lots of mechanisms of methylcobalamin. It directly protects cells against excitotoxicity. It has antioxidant and anti-inflammatory effects. It lowers the levels of a neurotoxic chemical called homocysteine, which that chemical in turn can induce oxidative stress, mitochondrial dysfunction, and excitotoxicity. And last but not least, it alters the composition of the gut microbiome. So this gets a trials grade of A because there's actually 2 pretty nice trials that were done in Japan. They were randomized, double-blind, placebo-controlled trials. They utilized a pretty high dose of methylcobalamin, 50 milligrams, delivered intramuscularly by injection twice a week. And they followed patients for up to a year, actually for longer than a year in some cases. And what they showed was a dramatic slowing in the rate of decline in ALS FRSR progression, 43%, and also a prolongation of survival by almost 2 years. with treatment with methylcobalamin. However, the qualifier here is in red. It only works if it started in the first 1 year of symptoms. There does not seem to be any benefit if it started after 1 year of symptoms. It's pretty safe. It's got a risk grade of B. The side effects are mostly from the injections, bruising, and pain. And it's mildly expensive, I would say, at a dose of 50 milligrams IM administered twice weekly. It's about $300 per month. Next, we come to nicotinamide riboside with pterostilbene. The commercially available form of this right now is called Basis. It's a supplement that's sold on a website from a company called Elysium Health. This gets a mechanisms grade of C. So nicotinamide riboside is a precursor to something called NAD, which is in turn a cofactor for lots of enzymes, including something called sirtuins. Sirtuins are believed to potentially affect multiple ALS-relevant mechanisms, including mitochondrial function, oxidative stress, and neuroinflammation. There's some evidence that NAD can affect the gut microbiome and, interestingly, suppress retroviruses as well. And the other ingredient that's in there, pterostilbene, that's an antioxidant that comes from blueberries. There's only one trial that's been published on this, and it gets a grade of D. It's a 32-patient, 4-month, randomized, double-blind, placebo-controlled trial. Now, they were using a product called EH301. It appears to have the same ingredients as Basis, but at 4 times higher doses. This is one of the only trials that I know of in the history of ALS that reports an actual improvement in ALS FRSR score. So on average, people that were being treated with nicotinamide riboside pterostilbene had an increase of 2.5 points on their ALS FRSR score over 4 months, whereas those on a placebo had a decrease of 5.5 points. It also reported increases in FVC and strength measurements in some patients. So it's a real eye-opener. It's one of those results that is almost too good to be true. I think it's important to recognize that there's many methodological problems with these trials. The two groups, the drug and placebo-treated patients, were not exactly equal after randomization. There was a pretty substantial dropout rate of 38%. There was a failure to use the intention to treat analysis that I talked about earlier. And then one of the authors of this paper is one of the owners of the product, which means they make money on it. That's a potential conflict of interest. The good news is there's another trial underway, which appears to be larger and using an even higher dose, and hopefully will address some of these methodological flaws. In the meantime, it does seem to be reasonably safe. I do have some questions about hepatitis and profound mental depression, which were experienced by 2 of the patients that were treated with the nicotinamide riboside pterostilbene in the trial. It really wasn't explained, how serious those were, but they weren't classified as serious adverse events. Rare GI side effects were seen in the trial. The costs of taking this, well, it will depend upon the dose. If you wanted to try to mimic what was done in the trial, you'd need to take 8 capsules daily. And it looks like on the website right now, that would cost you about $160 a month. And then last but not least is this one, which doesn't get a lot of attention, but I still think is quite interesting. This is tamoxifen. It's an oral estrogen receptor modulator. It's available by prescription to treat estrogen receptor positive breast cancer. So this actually gets mechanisms grade of A because it reduces markers of oxidative stress in humans. It can also reduce markers of neuroinflammation, stimulate autophagy, change the gut microbiome, and inhibit retroviral replication. So there are 3 small flawed trials suggesting that tamoxifen might be helpful. In the first one, 60 people living with ALS were randomized to receive different doses of tamoxifen, either 10 milligrams, 20 milligrams, 30 milligrams, or 40 milligrams, and they were treated for 2 years. And it looked like survival was significantly better in the folks who were treated with the 3 highest doses versus those treated with the 2 lowest doses. Criticisms here: small sample size, no blinding, lack of any real placebo control. The second trial was a selection trial. And here, 60 people living with ALS were randomized to get either tamoxifen at 40 milligrams a day, tamoxifen at 80 milligrams a day, or creatine at 30 grams a day over 38 weeks. People with ALS taking tamoxifen at 80 milligrams daily had a 50% slower rate of decline in muscle strength measurements, as well as a small decline in their ALS FRSR slope. Again, small study, no placebo control. And then last but not least, 18 people with ALS who didn't have SOD1 or FUS mutations were randomized to get either tamoxifen or placebo, and they were treated for a year. And the primary outcome measure was time to death or ventilator dependence. And it looks like the people who were taking tamoxifen were only half as likely to reach that endpoint at the end of the study as the ones on placebo. Now, that seems meaningful to me, but it didn't reach statistical significance, probably because we only had 18 people to start with. So I'm hoping that somebody at some point is going to try to replicate these interesting benefits in a larger, more rigorous trial. In the meantime, this gets a risk grade of D, and that's because if you follow people with breast cancer who are being treated with tamoxifen, they do seem to have a slightly elevated risk of developing other cancers compared to people with breast cancer who are not treated with tamoxifen. So it's possible that tamoxifen over a long period of time could cause cancer. Hot flashes are seen in 24% of people with ALS who are treated with tamoxifen. I will say this is, to me, quite a reasonable cost. 80 milligrams a day, it's only $80 a month. So that's the end of the list. And I want to just step back for a second and tell you about something that I think is kind of interesting. As I was putting this together, we struggle in this field to select targets for clinical trials. We get our ideas mainly from looking at cell models of ALS or animal models of ALS. And many of these, have potential flaws. Like, for example, our animal models are often based upon genetic mutations, which most people with ALS don't have. So it may not be all that surprising that when we develop drugs based upon those targets and we bring them into trials, most of the time they don't work. But this is sort of a different way of looking at targets. We're taking data that comes from people living with ALS, people that were in trials of things that most people don't pay a lot of attention to, these alternative or off-label treatments, trials that are flawed for sure. But to me, the data that comes from these kinds of experiments should be valued just as much as the data that comes from cell and animal models, which are 2 steps removed from patients. And so I find it interesting that these 8 promising products, they actually have some significant overlap in their mechanisms. 6 out of 8 target neuroinflammation, 6 out of 8 target oxidative stress, 6 out of 8 target the gut microbiome, 4 out of 8 target mitochondrial dysfunction, and 3 out of 8 target antiretroviral activity. And so I believe that these mechanisms should really be considered now to be promising targets when it comes to ALS therapy development. And certainly there are some companies that are out there developing products that hit these targets. So in the last few minutes that we have, I just want to remind folks about my approach, how I guide people who come to see me who want to experiment. So I think there's 3 parts to this. The first is to build a strong foundation of evidence-based care. There's a lot of things that we know now, both evidence and experience-based treatments that can slow the rate of decline in this disease, can reduce hospitalizations, can improve quality of life, and can lengthen survival for people with this disease. And so, most of the people that come to see me, they want to self-experiment, but it's interesting. Sometimes they come for the first visit and they're on, 30 experimental products, but they're not on Riluzole and they're not really going to any sort of multidisciplinary clinic. Like, to me, that's building the world's most beautiful house on top of matchsticks. You got to have a strong foundation of evidence-based care. Otherwise, the experimentation doesn't really make any sense. So the second thing I do is I talk about the different ways through which people could get experimental products. And I've got sort of a ranking system, which I expect is the same for most of us in this field. The preferred way to get experimental treatments is in clinical trials. Why? Well, there you're getting products where there's a lot of data behind them. We have a lot of reason to believe they're promising. You're getting products that we know are pure. And a lot of times when you buy supplements off the internet, we don't know about the purity. You're getting a lot of oversight in the trial for safety. You're getting a lot of things measured. So we're guaranteed to learn something from your participation in a clinical trial. So it's a win-win to me. The next best is expanded access programs. So we know that there's not enough trials. We know that the majority of people right now living with ALS don't qualify for most of the trials that are out there. So we need another pathway. And thankfully, expanded access programs exist and they're becoming more common. So these are FDA approved pathways through which you can get a product that's being developed in trials outside of the trial. And the number one thing is you can't qualify for a trial. So you can't be a person who could participate in a trial and just say, no, I'd rather be in an expanded access program. That's not allowed. But in these programs, you get the same products that are vetted, that we know about the purity of. You do get a lot of safety oversight. You do get some things measured. More and more, we're starting to see that we can measure things in group expanded access programs and we can learn. And so if there's no trial, if there's no expanded access program, then we're left with category 3, and that's self-experimentation. And that's where today's list of products probably has a role, the alternative and off-label things that are out there. And so for those who must self-experiment, my approach is let's add one product at a time, and let's start with the things that look most promising based upon our ALS Untangled reviews. So if you're coming into me and you're already on a large regimen, I'm also probably going to take things out that look like they don't work. So just as we have some products that have really great mechanism and trials grades, ALS Untangled also has, I think I counted last night, 16 products that have F grades for trials. It doesn't really make any sense to take a product that we've already studied in a trial and it got an F because it didn't show any benefits. There's so many options. Why take something from that category? I'll also probably remove products that have overlapping mechanisms. Like sometimes I'll see someone who's on 6 antioxidants. I don't think you need 6 antioxidants. So each time we start a new product, we're going to agree upon a target dose, hopefully a dose that's determined by previous trials, biomarker studies, safety studies, and we're going to agree upon the outcome measure that we're going to use and how long we're going to take it before we decide to give up on it. So right now, what I'm looking for is a 50% slowing over 6 months on the slope of the ALS FRSR. So somebody starts a new product, I say, all right, here's your slope coming in. Let's take a look at this. Once you get up to the target dose, let's have you on it for 6 months and let's see if we slow decline by 50% or more. And if we don't, then to me, there's not great evidence that this is helping you. Now, this is not perfect. We know the ALS FRSR slope is not a straight line in people. There can be spontaneous periods where it slows down, speeds up, plateaus, or even starts to reverse for a while. But for right now, I think it's the best we got. Something that I'm paying close attention to is the use of neurofilament light chain. I think there's going to come a time when we might actually be able to measure that at the start of a self-experimentation protocol. And in 6 months, look to see if that has decreased. The issue that I have with it right now is I don't really know what a significant decrease is. And so I do feel like if someone's ALS FRSR slows by 50%, that's meaningful and it's unusual in the natural history of the disease. And as I start to understand those parameters for neurofilament light chain, I'll probably start to incorporate that into my practice. So every now and again, somebody comes along and there's a product that we haven't looked at with ALS Untangled, or I get an email from someone about a new website that just sprung up the day before. And so I think this is where our ALS Untangled list of red flags can be helpful. So a few years ago, we went back and we looked at everything that we reviewed and we said, what are some of the things that are associated with the least promising products that we've reviewed? Those should be considered to be red flags. So if somebody wants to charge you a whopping sum for a product that's never been shown to actually slow, stop, or reverse ALS, that's a red flag. If somebody's out there advertising their product as effective against many of the world's worst diseases, which share no mechanism in common, that's a red flag. If somebody wants to give you something and there's no clear safety or scientific oversight, not good. if there's no informed consent, if there's no clear mechanism by which this intervention might help, if they want to charge you, give it to you, and send you on your way without ever measuring what happens, that's a huge red flag. If the person that's giving you this has no plans for gathering data and presenting it for scientific peer review at a meeting or in a journal, that's a red flag. You have to wonder, what is their motivation if they're not planning to present it? If the only evidence of benefits is anecdotes, stories of people that you can't find that reportedly improved on this, that's a red flag. If the person that wants to offer this to you has no relevant training, no track record of presentations at meetings or publications, that's a red flag. And maybe the biggest red flag of all is if the person who's offering this to you advises you to divorce yourself from your mainstream ALS doctor. Some of these folks that are out there portray themselves as victims of the system. I mean, the scientific method's been around for hundreds of years. It doesn't discriminate. Anybody who has an idea can use the scientific method to prove to the world that their idea works. So there should be nobody claiming to be a victim. And mainstream doctors have a lot of things to offer, which we talked about, that can help. So just to kind of summarize what I told you today, I feel like June 12th, 2024, there's 8 things that ALS Untangled has reviewed that look promising for the treatment of ALS. Now, again, they're not FDA approved. They're not absolutely proven to work. They all require further study. But I think it's fascinating that there's so much mechanistic overlap between these. And I think that should tell us something in terms of where to prioritize drug development. If you're thinking about something that's not on any ALS Untangled list, take a look at the red flags and see how many might apply. The more of those you see, the more you may want to avoid that particular new treatment. And I put up that approach because I hope that my colleagues who are out there who might be listening will use either that or some kind of approach. I mean, every person that I've ever met with ALS, almost everyone, has wanted to try experimental things. And so we have to have some sort of approach to guiding them. If we don't work with them, then they're going to have to go to the internet to find things. And again, the claims that are made there may not be reliable. So thank you to everyone out there who's living with ALS. Some of you have sent me emails this week and said, man, you're really giving a lot of talks this week. Well, you're the reason I stay up late at night and work on these and get up early in the morning and try to build, new patient-centric trial programs. I mean, you inspire me to keep going. I couldn't do it, no matter how much inspiration, if people didn't fund my work. So a lot of my work is funded by people living with ALS, families, foundations. Thank you for believing in me. Thanks to all the collaborators around the world, patients like me, Neurobank, Origent, all the 130 clinicians and scientists who work on ALS Untangled. This is a volunteer organization for them. And thanks to my care team at Duke for helping me build a great foundation of patient care upon which all my experimental ideas are based. So I'm going to stop the share now and take questions.
Hi, thank you, Dr. Bedlock, for another amazing and informative webinar. I do just want to let all of our listeners know that we've had a lot of questions. Will this be recorded? Trying to keep up on the information. It is being recorded, and we will have this on the NEALS website where the rest of the NEALS webinars are on neals.org. Hopefully today, we're going to try to get that up for you today. So Dr. Bedlock, we have a lot of questions coming in. I'm going to try to group them a little bit so we can get through as many as possible. First, one of the main themes of questions I see is we've talked a lot about the microbiome, and people are interested in your thoughts on the cases in Japan of the fecal transplantation that apparently significantly improved respiratory failure. And they want to know what you think of that and why there aren't more trials like that here.
Yeah, so these are fantastic questions, and I'm smiling because I've been trying to sort out the gut microbiome in ALS for a few years. If you go back through the archive of recorded NEALS webinars, you'll find one that I did, I think it was 3 or 4 years ago, called "Are We Flushing the Key to ALS Down Our Toilets?" I actually do now believe there is something in the microbiome that's important. And so my colleagues and I here at Duke, with the help of our patient partners who funded these studies, people at the Martha Olson Fernandez and Foundation and Augie's Quest, we've learned that there is a difference in the composition of the microbiome between people in our clinic who are progressing slowly and people in our clinic who are progressing fast. Unfortunately, the difference is not simple. It isn't one organism that we can just take and put into a probiotic and change the gut microbiome and slow ALS progression. So we've also done some experiments where we take the stool from slow or fast progressing patients in our clinic and we put it into our genetic model, our mouse model of ALS, and we can change the progression. And so right now I'm working with two groups. One group that has a probiotic that's close to what we think we need to do, and also with the Duke fecal transplant team. There actually is a whole team here that does fecal transplants for FDA-approved indications like infections in the gut. And we're trying to figure out how we can get one of these trials off the ground. And we may wind up doing both because we don't know what the best approach is. But as we've been working on this, there have been 3 case reports, 3 in the past year, 2 that you just mentioned, but there's also one that's previous to that, where it looks like a person with ALS who was progressing pretty quickly got a series of fecal transplants from a healthy donor where the stool was screened for infections. And it does look like there was a significant positive deflection in the rate of decline on a variety of different validated outcome measures. So I think that strengthens this idea that manipulating the gut microbiome, can slow the progression of ALS. There has been a trial listed on clinicaltrials.gov for a couple of years. It says that it's doing fecal transplants. I haven't seen any results. I haven't seen anything presented at meetings, so I don't know where that trial is, but certainly here at Duke, we are going to be doing 1 or possibly 2 different trials in the next year to manipulate the microbiome and see if we can slow ALS progression.
Amazing. So in some of your overviews, you had mentioned the microbiome in the context of it affects the microbiome or it may have impacts to the microbiome. And there are some questions about what did you actually mean by that?
Yeah, so in every case where I mentioned that, there's evidence from either an animal model or from another human study that product changed the composition of the gut microbiome. Now, again, the problem is this is not simple. There are actually trillions of organisms in the gut microbiome. Like when you get a readout of the microbiome composition, it's this massive grid of all these different organisms that light up different colors depending upon how much of that organism is in there. And so I wish it was simple, like, giving L-serine boosts Saccharomyces. Man, I wish that was that simple. And that's part of the struggle that we're having, is trying to figure out how to reconcile these massive changes that you see with what we think might be a good and bad microbiome. And that's part of why we're struggling a little bit to figure out what the best probiotic might be. But I think it's better to take a shot with what we know than to sort of wait around for years trying to figure out what's perfect. I think somebody once said, don't let perfect get in the way of doing something good. So that's what we're going to try to do. But again, it may be that what we need to do is something more global, like rather than try to change one organism or even a small handful, we might need a big change like with a fecal transplant. And then the question is going to be, what's the best source of the stool? Should it be a person without ALS, or should it be a person that has very slow progression? So that's another thing that we're trying to grapple with right now.
Fascinating. And, so many levels of things brings us to the next topic. There are so many potential off-label or alternative therapies that people are interested in trying, and there's confusion of how many should I take? Is it okay to take more than one? And then there are questions of, well, Dr. Bedlock, why did you say you take people off of them? Isn't more better? So if you could talk a little bit about that.
Yeah. So again, I think for me, there's 2 approaches, right? Sometimes people come and they're just on one thing or not on anything. They haven't tried anything experimental yet. And that's much easier, because then, if they're not a trial candidate, they're not an expanded access candidate, then I can say, well, look, here's a list of 8 things that today I believe are the most promising things you can get your hands on. And, based on other characteristics of your disease, do you have bulbar dysfunction? How far into the disease are you? Are you within the first year? we would use things like that to decide what might be the most promising first product for you to try. And again, the reason I would say you try one product at a time is we really don't know how all these things interact. And that's one of the things I worry about when I see someone literally coming in with a hefty bag full of stuff. I mean, I think the record I had was 90 supplements that somebody came to me on. I have no idea how all that stuff interacts, much less how it interacts with, the medicines that we use, Riluzole, Radicava, medicines for spasticity, or pseudobulbar affect. There's no way to figure out an interaction when there's that many things going on. So I much rather try to keep it simple. But the reality is sometimes people do come and they're on a whole bunch of these things. And that's where I take the approach. First, let's get rid of things that we graded as an F for trials. Next, let's get rid of things where there's an overlapping mechanism. So don't take 6 antioxidants. Like, that doesn't make sense to me. So let's try to pare the regimen down and let's add one thing. Let's see if there's something, in that list of 8 promising products that hits a mechanism you're not hitting with your regimen. And again, we try over 6 months to say, what are we seeing? What's happening? Are we seeing any slowing? Sometimes I have people that come and they say, oh, I took that for a week. Oh, I took that for 2 weeks. Didn't do anything. Well, if you think about the biology of ALS, if we stopped whatever was killing the motor neurons, you probably would need 6 months to start to show some recovery of lost function. It isn't going to happen overnight, because the way that you would recover lost function is your surviving motor neurons would send off branches, and those only grow at a millimeter per day. So, I mean, if they got to grow 3.5 feet to get down to where your big toe is for you to be able to wiggle that again, that ain't going to happen in 2 weeks. So I think for each new thing that we try, that's why I sit down and I say, we got to have a plan. If we're going to do this together, we got to agree on a plan. Like, I don't want to hear when you come back that you bailed on that after 2 weeks because it wasn't working.
Yeah, that makes perfect sense. So is there a way to generalize all of these alternative therapies, the effectiveness or potential effectiveness? Does it matter where in the ALS experience the patient is and how progressed they may be?
Yeah, so I would say I do use that information to try to decide which one of these I think is best for a patient. So just to repeat what I said earlier, like if you have bulbar dysfunction, speaking problems, swallowing problems, drooling problems, well, I think Nudexa is probably something reasonable to consider. And if you have pseudobulbar affect, we might even be able to get it paid for, because there isn't any other of those 8 products that seems to improve bulbar function. If you have already lost your independence, acetyl L-carnitine might not make sense, because that's the main thing that was seen in that one clinical trial, is it preserved a person's ability to feed themselves, dress themselves, adjust their covers, and walk. If you've already lost all that, I don't really have any evidence that that medicine works for you. If you're more than 1 year into your weakness with ALS, well, methylcobalamin probably doesn't make sense then, because the trials that were done don't show any benefit for people that had symptoms longer than a year. So these are the kinds of details that I'm trying to ferret out as I try to personalize my approach to which product we should try first.
I love that. This is a very interesting question. I'm wondering myself, what is the difference between clenbuterol and albuterol?
Mhm. They're in the same class, so they're both considered to be beta agonists. Clenbuterol has a much longer half-life. And so, when we're trying to pick a drug out of a class, part of the promise of the drug is going to depend upon how long it stays around, how well it gets into the brain, what we call the pharmacokinetics of the drug. And so, as we were trying to figure out what to study, to us, it looked like the pharmacokinetics of clenbuterol would be much more promising for a disease like ALS than a shorter acting agent like albuterol.
That makes sense. I know we're right at the top of the hour, and there are a number of questions that have come in where people are asking personal questions about their situation to you. So we're not going to be able to take those live right now, but I encourage them to reach out to their care providers or to you, Dr. Bedlack. One question I do want to ask you before we end is, people are asking, what are you most excited about in the science of ALS right now?
Wow, so many things. I mean, if I had to pick just one, Allison, I would say what I saw Dr. Chaikovitz present in Basel, Switzerland, it's still, it's when I lay in bed at night and I think about the things I'm thankful for, that presentation like just exploded the amount of optimism and hope that I have, because even though tofersen is only for 1 to 2% of people with ALS who have SOD1 mutations, we understood the biology of that form of ALS really well. We knew what we needed to do with a drug to help people with that. And now that we see that people have been on this drug in some cases for up to 2 years, Dr. Chaikovitz showed that about half of the people on that drug for a long period of time have stopped progressing, and about 20% are getting stronger. It's the first time in all the years I've been going to ALS meetings where people stood up and gave a standing ovation at the end of the presentation, because it changed everything. I mean, it changed our concept of what's possible if we understand what we should be targeting and we find a really good way to target that. We don't have to talk about a 30% slowing in decline. We could talk about stopping or reversing decline. And so, I mean, that's what I'm after, in the who knows how many years before I retire, but that's what I want to see is more of that.
I hope you have quite a few more years, because your hope is contagious. So thank you for all you do for our community. Want to let everybody know who's on as our listeners that we do have 2 upcoming webinars, one on June 17th about respiratory care and one on June 25th about genetics in ALS. So we are keeping going with information. I want to thank all of our listeners for joining, and Dr. Badlock, a special thank you to you. Thank you for taking the time to share that information with us and for answering my questions. That's one of my favorite things to do in my whole life, is ask you personally questions. So thank you for doing that, and I hope everybody has a wonderful evening. Thank you.
Thanks, everyone. Bye-bye.